ESM-2 is a protein language model. It learns biological patterns directly from protein sequences. Our goal is to predict protein function. We start with proteins and known labels. These examples will teach our classifier. ESM-2 is a pretrained transformer. It already understands protein sequence patterns. ESM-2 first converts amino acids into tokens. Special tokens mark the beginning and end. The transformer analyzes relationships across the protein. It can connect amino acids that are far apart. ESM-2 creates an embedding for every amino acid. These vectors capture biological information. We average all residue embeddings. This creates a single summary vector for the whole protein. Each protein becomes a row of learned features. This table can be used by machine learning algorithms. We train a classifier using ESM-2 embeddings. The model learns the difference between enzymes and non-enzymes. A new protein follows the same pipeline. The classifier predicts its function. First, the protein sequence is converted into tokens. ESM-2 transforms each amino acid into learned residue embeddings. We average those embeddings to create a single protein representation. Finally, a classifier uses that representation to predict the protein's function.